everybody. It is my distinct pleasure to introduce Dr. Emery Neil Brown today. We have been colleagues for many, many years, and we share the same field of hippocampus coding and also sleep and anesthesia. I was in the anesthesia department for 15 years, and Dr. Emery Brown has an MD, PhD in anesthesiology and statistics. And I was wondering, because I don't know him all that well. I really like him, and I've really admired him. I've seen him give really big talks, but we haven't had that many deep conversations, and I've wondered, why is that the case? Because we share two, at least, major fields. And when I took a deep dive into his CV in order to prepare for this introduction, I realized why. The man is a machine of productivity, and in the past 20 years, my 55 papers have been matched with his 288 papers. He published, as I calculated it, 14.4 papers a year. That's one every 3.6 weeks. And in 2012 alone, I just picked a random year, he published 30 articles. That's one every 1.7 weeks. So, this guy is busy. Not only is he a practicing anesthesiologist, an actual anesthesiologist, Mark Baxter here said he's the only man that he would actually trust to put him under. So he's actually a really good, trustworthy practicing anesthesiologist. He's also a mathematician and a researcher that has touched so many different fields. He helps people analyze their data and come up with more sophisticated ways to look at the data. And so in all of these papers, he has really pushed forward multiple fields. His own field is more, he would say what he owns in his own laboratory is more related to, especially these days, anesthesia and the coding of different states and helping people, helping anesthesiologists through a closed loop system determine that looks at EEG, determine how deep and what kind of anesthetic state they're in and which anesthesia they might need more of, because now anesthesiology practice utilizes many pharmacological agents to induce the lack of sensation, the unconsciousness, and the immobility. And there are millions of people every year, hundreds of millions of people every year that get anesthetized for various procedures, and he's really helping all of those people come through with flying colors instead of doing it blind like they've done it throughout history. Dr. Brown was born in Florida. He has two older brothers. His parents both taught high school and middle school mathematics, so they kind of pushed math on him, and he didn't like it very much until he reached high school and college, when he realized how powerful mathematics really was in answering the questions that are posed in the world. He went to high school at Phillips Academy, Exeter. I went to high school in Phillips Academy, Andover, which is kind of like the rival school, so we really do have a lot of parallels. He was enthralled with the Romance languages and thought maybe he would be a Romance language major in college. And he went to Harvard and started there in 1974. But his roommate there was really interested in economics and applied mathematics, and influenced Emery to, in fact, switch his major to be an applied math major. He spent 6 months in Spain and another year abroad in France. And then he went back to Harvard for his MD, PhD, and his PhD was in statistics. So he did an internship at Brigham Women's Hospital, a residency in anesthesiology at Mass General Hospital, and since 1992, he's been at Mass General Hospital as an anesthesiologist. In 2005, he got a joint appointment at MIT, so he's both Harvard and MIT, and there he's the Edward Hood Taplin Professor of Medical Engineering and Computational Neuroscience, and at Harvard, he's the Warren M. Zappel Professor of Anesthesiology. So his three fields that he's particularly influenced is the first field he was in circadian biology, And his first paper with Chuck Seisler has been published, cited at least 763 times. His second big field is neural decoding and statistics that helps, for example, place field and hippocampal place field people to figure out how to see what's significant. And then this third field is anesthesia state characterizations. He's trained over 40 trainees, all of which have gone on to really glorious careers of their own. And he's gotten a Director's Pioneer Award at the NIH. He was part of Obama's Neuroscience Dream Team in 2013 and 14, part of the Brain Initiative Working Group. He's on the board of directors at the Simons Foundation and the board of trustees at the Guggenheim Foundation, and he's one of only a handful of people elected to I was going to say 3 academies, but actually now 4 academies: the National Academy of Medicine, the National Academy of Science, the National Academy of Engineering, and the National Academy of Inventors. And he's the first African American to be part of all of these societies, the only statistician and the only anesthesiologist to be part of all this. All of this, in addition to the fact that he still has a big heart, like his parents did, in allowing young people to get excited about mathematics like he has been able to employ it so well in his life. So he's a big part of something called the Calculus Project, which helps middle schoolers, especially from age 6th grade on, especially students from underrepresented backgrounds like he is, to develop a love for math and to be trained in it early on so that they can really succeed like he has. So all of this to say, I'm just really, really pleased to know Dr. Brown, and joining us from home where he is undoubtedly working on 3 papers simultaneously. Please join me in congratulating and welcoming Dr. Emery Brown.
Gina, thank you very much for the kind introduction. And Michael, thanks so much for the invitation to speak here. It's a real privilege. And again, I sincerely apologize for not being able to make it there in person. But I was really very much looking forward to participating. It's a fantastic conference, and sorry I'm missing it in person. I want to talk about today monitoring and controlling unconsciousness under general anesthesia, because I thought this is a very appropriate topic for learning and memory, because I want to show you how we've learned the various dynamics of the anesthetics. In particular, I want to focus on propofol and how we can actually use that to create better control of anesthetic states, and also turning the brain on and turning the brain off. So that's what I want to talk about today. So these are some of the sources of funding that I've had from the NIH, my Department of Anesthesia, as well as my institutes at MIT. And this is the outline I'm going to follow. I'm going to give you a clinical look at general anesthesia. We'll talk about unconsciousness maintained by propofol and other anesthetics. I want to talk about aging in anesthesia. Then we'll get to closed loop control, and finally reanimation, turning the brain back on after anesthesia. So here are the main points that I want to make. I don't know what concept you have of anesthesia right now, but it's not a static state. The brain is dynamic. It's not turned off. And one of the primary ways that the anesthetics work, that they create these states of altered arousal, in particular unconsciousness, is by inducing and sustaining oscillations. These oscillations impair the various communication among the various brain regions. I'm going to show you that in just a second. They're easy to see on the EEG. They change systematically with anesthetic class, dose, and also patient age. So that means that it provides a way to monitor the brain state of the person under anesthesia. Closed loop control of anesthetics is very feasible. It's not a practice in the United States. It's been done experimentally in Europe, and this is something which we're pushing forward right now to try to bring to fruition. And then the final idea is to turn the brain back on. As I said, reanimation. We give you drugs to put you in a state of anesthesia, general anesthesia. Then what we do is we just let the drugs wear off and let you wake up passively. And not surprisingly, particularly older patients and often kids have brain dysfunction after anesthesia because the effects of the drugs are still there and linger. So we're going to talk about what we call reanimation, or turning the brain back on. So let's take a clinical look at general anesthesia. So what is general anesthesia? So a drug-induced reversible state that consists of anti-nociception, so that's control of nociceptive processing, unconsciousness, amnesia, akinesia, and physiologic stability. And so the reversible is important and the stability is important, because if you take these four things here by themselves, they're synonymous with death, and that's not too cool. It's often said how anesthesia works is a mystery, but it actually is not a mystery. We're going to show you, as I said, the drugs create oscillations that impair how the brain communicates. That's one of their primary mechanisms. There are other mechanisms, but that's a primary one. So how do we actually create the anesthetic state? During maintenance, during a time after you've induced anesthesia, you use combinations of different drugs to create the anesthetic state. So propofol or the inhaled drugs are all still ethers. So isoflurane, sevoflurane, and desflurane. So you could use them to create all four of the states just alone. But what we do is we use combination of drugs. So for the antinociception, we use the opioids. Unconsciousness, we induce with propofol, we may with the inhaled drug or with propofol. Then we give muscle relaxants, anticholinergic drugs to create immobility and reverse that in the case. So the idea of using combinations of drugs, you get most of the good effects and fewer of the side effects. That's the idea between what we call balanced anesthesia for maintenance. As I said, what we do is at the end of the case, we just let you come to. We don't give you something to turn your brain back on. So let's talk about unconscious maintained by propofol and the other anesthetics. So I'm going to show you a little video here. This is a video which was made almost about 10 years ago now, of a woman that was taken care of. She was about 60 years old. She weighs about 80 kilograms. She's going to have thyroid surgery. I'm going to give her a bolus of propofol. I'm just going to play the video and I'll talk you through it. So 4 leads of EEG, which we can put on all of our patients if we take them in the operating room. And what you're going to see in a second is it's going to get very noisy. And it's going to get noisy because when you inject propofol into a small vein, like the one on the back of my hand, it burns like crazy. So this is going to start burning like crazy. So, and that's her picking up right there with the propofol. And that's actually going to be good from a pedagogical standpoint, because the moment the drug takes effect, you're going to be able to see 3 seconds. Watch. 1, 2, watch here. Theta oscillations. Kind of get deeper. Slow oscillations. Even deeper. Now she's going to go into burst of pressure. You're going to go flat, then burst, and then flat. So what did I just show you? So she was like this awake. So these are sort of like gamma oscillations, 5 to, let's say, 5 microvolts or so. And then when in the sedative state, you saw her go here, beta oscillation. This is the state I try to keep her in. She's going to have a colonoscopy, and I just wanted to have her so she's arousable but not completely unconscious. Then you saw her go to these large, slow oscillations here, like, between about 0.1 to about maybe 2 hertz or so. And this usually indicates the time that the drug is hitting the brainstem. You can actually see it if you check the brainstem signs, you'll actually see that. Then she went to this deeper state, which was burst suppression. This is a state of profound brain inactivation. And keep this in mind, because we're going to use this. We're going to actually, this is going to be one of the states we're going to control later on. And between the bursts, she's actually isoelectric. We skipped over this state here, the slow and alpha oscillation. This is the state we'd like to maintain her in for surgery, because she's sufficiently unconscious. It's not too unconscious, maybe down here, but she's sufficiently unconscious so that she can undergo surgery, but not so much that we're sort of overdoing it. And we'll come back and take a look at that in a second. So what happens from here? Look, I just want to draw your attention to this. You go from 5 microvolts, like, down to about 20 to 50 microvolts, and in kids, we'll see in a second, there's 1,000 microvolts. So you have these oscillations changing from like small amplitude, high frequency, to large amplitude, low frequency oscillations. And it's not a subtle change. You just saw it take place. That's a very typical induction that we see every day in the operating room. And so, but if we look at this in the frequency domain, it's actually easier to see it better. So if I come back here, I did the same thing with this 19-year-old woman I was taking care of a few years ago. Gave her an induction dose of propofol here, where the arrow is. And then she went into the slow oscillations. Then she converted into showing the alpha oscillations, the 10 hertz oscillations, which you're showing here. And then the slow delta oscillations going from 0.1 to about 4 hertz. This is a classic signature of propofol. And I maintain this by maintaining on infusion. This is a spectrogram. So by breaking the signal up into its spectral components, it's much easier to see the anesthetic state. and sort of appreciate the state in which the patient is in. So here's the alpha oscillations and the slow delta oscillation. So as long as I keep and you can see them up here in the time domain, see the high frequency ones there, that's these, and the lower frequency ones, which are like here, like that, that's the ones down here. So as long as you maintain the infusion, she's going to stay in that state. You turn it off, they break up, and she eventually wakes up over here. So this is a classic signature of propofol or any GABAergic anesthetic. So propofol works by enhancing GABAergic inhibition, just like sevoflurane, for example, or the inhaled ethers or the barbiturates. So these are my colleagues that I've done this work with over the last several years. Patrick Purdon at MGH, Nancy Gopel at Boston University, Shenung Ching, who's now at WashU, Maud Eskandar, neurosurgeon now at Einstein, Eric Pierce, anesthesiologist at MGH, Laura Lewis, former PhD student, who's now in the faculty at MIT, and Sid Cash, neurologist at MGH. So let's go back and look at those oscillations. So remind ourselves of a little bit of anatomy. So we have the GABAergic effects of propofol are going to be seen most prominently at the interneurons. So there are cortical interneurons, three areas I want to point out. Cortical interneurons, let's say around the thalamus, the TRN, the thalamic reticular nucleus, and also these inhibitory inhibitory brainstem neurons, which come out of the preoptic area of the hypothalamus. So propofol goes to all these places as soon as it enters the bloodstream. And so, but what do the patterns tell us? So this is the sort of pattern we see on the monitor. And this is not an enhanced picture, what have you. This is exactly, it looks just as clear, if you would, on the monitor in the operating room. So let me tell you one little factoid. You see this alpha oscillation that I'm showing you here. It's actually concentrated in the front of the head. And what we figured out is we think it's an oscillation. Our research, mathematical modeling, and also experiments in rodents show that this 10 Hertz oscillation is going back and forth between the thalamus and the cortex. And it's making it very difficult for these two areas of brain regions to communicate. I'm going to show you that in more detail in a second. Then we'll talk about sort of the slow oscillation and the slow delta oscillation. But you've got these, so this thalamocortical oscillation here. Then you have the slow oscillations, which are sort of over the entire cortex. And the thalamocortical oscillation is just concentrated in the front. It's not in the back. So it's sort of like this. So this is a person awake. They're conscious, like they have the low frequency, high frequency, low amplitude oscillations. And there's sort of like broadband communication between thalamus and cortex. When you give a drug like propofol, which slows all this down, and you have these 10 Hertz oscillations, which you can see right here, and the slow oscillations, which make it very difficult for the person to for the parts of the brain to communicate, particularly the thalamus and cortex, which are in this 10 Hertz loop. So this is one of the ways that you can see unconscious occurring. Let's look at the slow oscillation. So that's the 10 Hertz oscillation. The slow oscillations are across the entire cortex, and this is work from Laura Lewis's PhD thesis almost a little over 11 years ago. So these are recordings of LFPs from patients that have epilepsy, who've come for epilepsy surgery. So they get grids and strips implanted. They stay in the hospital for 5 to 7 days off medications. And then once the team has figured out where the seizure foci are, they come back and have the grids and strips removed. And then they subsequently come back for surgery. So during that time when they come back to have the grids and strips removed, you have an amazing natural experiment. You have a human with electrodes in his or her brain that you can record from and see what neurons in the human brain do during anesthesia. And so this is what it looks like. So this is the person that is conscious. And you see the LFPs, and they correspond to these areas here on the medial temporal lobe here. So this is a neuron which is next to the red electrode. It's spiking pretty much wherever it wants to. Now look what happens when the slow oscillations start. So this is a slow oscillation occurring when the person's unconscious. You can spike here, you can spike here. So you can see the spiking is now spaced out every 2 seconds or so. So you can imagine if you can only spike every 2 seconds, it's going to be very difficult to be conscious if that's happening across the entire cortex. And to get a better look at the cortex, we did the same experiments with Earl Miller and his postdoc, Andre Bastos, and Jake Donahue, all at MIT. And so these are 4 brain regions. So these are 2 frontal areas, area 8A, the frontal eye fields, prefrontal cortex. There's the posterior parietal cortex and superior temporal gyrus, so the auditory area. So two kind of posterior areas and two frontal areas. And here, so these are the oscillation when the animal's awake, non-human primate. And then if you look here, here's what the spiking activity looks like. You can see the neurons are spiking pretty much wherever they want to. And the spike rate is about 10 to 12 spikes per second. Now here's the same thing under propofol, just like we did in the human case, where now they're the large slow oscillations. Now look across the brain regions here. There are these little islands now, but you can see these very large up-down states. So spiking here, no spiking, spiking, no spiking. And they're lined up in a very nice way across the entire, at least these four cortical regions. And the key thing to appreciate is that the spike rates have gone here from 10 to 12 spikes per second down to 1 to 2 spikes per second. So like a 90-95% drop in spike rate. So when you see the slow oscillations on the EEG monitor, if this is occurring, then you can know the person is profoundly unconscious. You can actually see why. So I've shown you the alpha oscillations, which I've suggested is strongly coupling between the thalamus and cortex, and the slow waves, which are creating these islands, preventing the areas across the cortex from communicating. I didn't talk about anteriorization, and I showed you the brainstem effects on induction, the large slow oscillations which come on right during induction. That's primarily a brainstem effect. We can infer that clinically from doing a clinical exam on the patient at the time of induction. So we went into a little bit of detail here with propofol. We could do the same thing with each of the other anesthetics. And even though you can see the differences perhaps in the time domain signatures here, if you look at them in the frequency domain, it's much easier to see. This is ketamine, which is really has been an enigma in a lot of instances because instead of the EEG slowing down, it actually speeds up. So this is low dose ketamine, the same sort of dosing that the psychiatrists use when they're treating patients with depression. And then sevoflurane, which has a pattern similar to that of propofol, which isn't surprising, because even though this is a gas and this is a IV anesthetic, they both work primarily through GABAergic mechanisms. And the ethers have an additional oscillation, a theta oscillation, which we still haven't deciphered where it's coming from. And dexmedetomidine produces slow waves, which are very much akin to the slow waves you see in slow wave sleep. So this is something we're trying to teach anesthesiologists to do so they can use to actually monitor the patient state much better during surgery. And we can, just like we did a bit of a deep dive into the circuitry of all the propofol, we can do the same thing with sevoflurane. And we've done this with sevoflurane, with ketamine, and also with dexmedetomidine. So the oscillations, the key thing is the oscillations are not an epiphenomenon. They're actually part of the mechanism through which the drugs are acting to create altered states of arousal, in particular unconsciousness. So let's look at the anesthesia and the aging brain. So this is what I showed you earlier. This is a young person in some 18 to 35, let's say, getting propofol, alpha oscillation, 10 to 12 hertz, 8 to 14 hertz, and then maybe slow delta oscillation from 0.1 to 4 hertz, classic picture, which I've shown you several times already. And then here's the same thing in a person who's 57. You can see it pretty clearly here, but a little weaker, maybe slightly lower frequency bands, but still they're quite visible. And if you look over here, here's someone who's 81. This is a lady I took care of who had a tumor on her chest the size of an American football. It took the thoracic surgeon the better part of 6.5 hours to remove it. But I was able to keep her well anesthetized with minimal dose of anesthesia, giving her 1/3 of the already age-adjusted dose of anesthesia. I was watching her EEG. And not only are the oscillations weaker, but notice the profound absence of oscillations here in the cognitive range. I mean, this is on a decibel scale, so this is about roughly a 25-35 dB change in the oscillations here. And these two guys are about the same age. His EEG looks like hers. His EEG looks like his. We age different physically. It makes sense that we age different, that our brains would age differently as well. And so and at first it might seem a bit surprising that, you would see this under anesthesia. But if you think about anesthesia for the purposes, not for anesthetizing someone, but you think about using it so that you can do just a neuroscience experiment. You put in a stimulus and you measure response. So you put in a stimulus, and the stimulus you put in is the anesthetic, and the response are oscillations. So if the brain is nice, young, and healthy, it can oscillate like this, but as you get older, it oscillates like this because the neurons are aging. And you can just run down the list of things that are changed with that aging. The mitochondria, are not as effective. Cell volume changes, the myelin sheath breaks down, the dendrites don't extend and retract as much. You don't have as much neurotransmitter being released. And if you go back to younger brains, you can just see this is a 3-year-old, this is a 14-year-old. They have the same patterns across time, but they're much broader spectral, much broader spectral content, and meaning much more larger oscillations spread over a much wider frequency range. So when you build algorithms that work to control or to monitor the brain here, they don't apply automatically. In fact, they often don't work in kids because the kids have similar dynamics, but across a wider spectral range. And this is one of the reasons that a lot of it wasn't appreciated that these monitors that were built to take care of adults weren't working in kids, what the reason was, but it's very obvious when you look at the spectrogram to see this. So let's talk a little bit about closed-loop controlled general anesthesia. So these are 3 very well-known people who had head injuries, and as a consequence, they had to be placed in a medical coma. So what? Something extra seemed to be. Oh, I see. Malala's name moved over here on top of my other text. Sorry about that. So what is a medical coma? Basically, a medical coma just means that you give someone a large dose of the word is thrown out a lot during the COVID pandemic, but what the real medical coma is you give someone a drug like propofol, and you place them in a state of profound anesthesia in order to allow the brain to rest, either to decrease intracranial pressure or to sort of take care of, or to treat sort of seizures which are unresponsive to standard therapies. So this is often done just by titrating the drugs manually. It's not done in any sort of controlled manner, and it's done by essentially a lot of guesswork. You say basically what sort of pattern of burst suppression that you'd like to see, how much suppression you'd like to see, and then you try to titrate the drug to do that. So, but we thought that this would be a perfect place to build a control system. So this work goes back 10 years ago. So Miriam Sinechi was one of my PhD students and postdocs, and Xiang Cheng did this work back about 10 years ago. So you did it in mice, in rats. And so here's what you do. You measure the EEG in real time. You have a target that you want to hit in terms of the pattern of burst suppression you'd like to see. You find out what the estimated target is. The difference between the estimated target and the actual target changes the infusion rate every 5 to 10 seconds of propofol going to the animal. And you measure the EEG and you continue this loop like this. And so here's what you're looking at. So here's burst suppression. And it's easy to find the target because burst suppression can be characterized on a second-to-second basis in terms of the fraction of time you spend suppressed. So in 1 second, what fraction of time are you suppressed? The way I've drawn it here, this is 60% of the time. So if this is 1 second, so there's suppression here, there's burst there and burst there. So this person at this point is like 60% suppressed. So you could set that as your target. So that's a target that you would set here, and then you would try to maintain that to generate the state of a medical coma. And it seems very logical you'd want to do something like this because people have to be anesthetized for several days to be in medical coma, as happened during the COVID pandemic. It makes sense you should try to do this with the help of a computer instead of manual. So let me just play this video just to show you this. So right here, here's the target. So Miriam starts it at 0.4. Here's what the controller is doing. This is the EEG up here. This is the infusion rate of the pump being updated every 10 seconds. So here's the target, 0.4. She's going to push it up to 0.7 next. So she pushes the target up to 0.7, and time on this axis is in minutes here. You see the controller is actually holding on to the 0.7 actually very tightly. Now she's going to push it up further. She's going to push it up to 0.9. So 90% suppressed. So meaning taking the pressure even deeper. So there it is, 90% suppressed. And the controller has no trouble doing that. So you can see that this is very feasible. And in fact, in this paper that she published, again, this was 10 years ago, this is the case I showed you, so going up, going up, going up. Here's a case, these are like six different experiments, 6 different animals, where here you maintain it here, you increase it. Now to decrease it, what you have to do is you have to turn the controller off, because there's no drug to make the sort of the rate of burst suppression go down. So the controller turns the infusion off, and then once the level drops to where it wants to be, it kicks back on again. So you can see she did this over a wide range of sort of configurations. So this is highly feasible. So we did that, and the FDA said, well, that's nice and cute, but you have to do it in larger animals. You have to do it in something which are going to be close, animals that are closer to humans. So in the last few years, with the help of Earl Miller, again, who's a very well-known non-human primate neurophysiologist, we built the same similar system. Suresh and Jake Donahue. Suresh was one of my postdocs, and Jake, as I said, was one of our PhD students, using the same concept. So again, a controller, a marker that you set a target for. You estimate where the target actually is. Difference changes the infusion rate. Now the duty cycle is 20 seconds now, as opposed to 5 to 10 seconds, and it's going into the non-human primate. And it just cycles through sort of very standard control setup, nothing fancy here. Excuse me. But here's the bit of biology which I think is really important, the neuroscience which is important. When you give a constant infusion of propofol, remember I showed you how when those raster plots of how the spike rates had dropped dramatically. So we're not going to have electrodes in a monkey's brain to be able to do control. We're not going to be having electrodes in a human brain. But we can measure LFP and eventually in humans EEG. And so here's the biology that makes this go. The spike rates dropping are tightly correlated with the animal becoming unconscious. And that actually correlates well on the order of about 0.9 with the power in the 20 and 30 hertz band of the LFP and eventually the EEG. So if we monitor the power in the LFP, we can have a signal that we can control and actually keep the animal in whatever state of unconsciousness that we want. So let me show you this now for non-human primates. So Suresh set his marker, so it should be at 1 first. And here's the infusion rate of the pump. And the first thing that he's going to do is he's just going to let the propofol run at a constant rate. This is what we do in the operating room. So this is going to simulate what we actually do in the operating room now. Then after about 30, 35 minutes, he's going to let the controller take over. So there's the marker. It's not at 1, it's wandering all over the place. You see the infusion rate is just a little bit below 0.4. So it's not controlling it very well at all. Now he's going to turn the controller on, so the controller takes over and it locks the marker in right at 1, right where you want it to be. Now what he's going to take it to a lower number. A lower number means a deeper state of anesthesia, unconsciousness. So he brings it down, so it drops down. And then in the final about 30 minutes, 20 or 30 minutes, he's going to take it higher, meaning to a lighter state of anesthesia above 1. So it's totally feasible to do this sort of control. So in other words, and I want to point something out here, which is really important clinically. And again, time on the axis here is in minutes. So look at this right here. This is someone in the operating room. This is someone in the operating room, the way this works now. Someone's getting a constant infusion of their anesthetic, and they're just getting deeper and deeper and deeper. And what did the controller do? The first thing it did was it actually shut off for 3 minutes so that it could get back up to where it needed to be. And then it holds the marker right where you actually want it to be. So once you see this and you realize that EEG is not commonly used, or it's not standard of care, and you realize that even if they were using EEG, you'd have constant infusions of propofol, and you're generating these large oscillations, and they're on there for 3 to 4 hours, if that's how long the surgery is, you can understand why brains wouldn't work afterwards. It makes perfect sense. It's no mystery. So the controller would help really dose the drugs much more precisely. So we have to, complete the studies in non-human primates to satisfy the FDA requirements to sort of test the system out in humans, but this is very feasible, as I've suggested here. So let's talk about reanimation as a final topic. So this is work that I've been doing with my colleague Ken Salt at Mass General Hospital. And this is a rat that Ken has anesthetized with propofol. He has an IV in his tail vein. And like I said, we don't give you anything to turn the brain back on. We actually just let you come back. This animal has an IV in his tail vein. He's getting propofol. So he's anesthetized. And he also has an electrode in his ventral tegmental area. So in the top of the midbrain, an arousal center, very important for cognition and also reward. So the stimulation is off. Ken's going to stimulate his ventral tegmental area. And watch what happens when he starts stimulating. Look how he starts breathing like crazy. The propofol is still running. So whatever is taking effect is counteracting the effects of the propofol profoundly. Now, he gets up and he runs off the table. All right. So he's awake, all right? But is he cognitively processing? So Ken did another set of experiments, which he trained a rat to actually do a task, which he hits the correct light, turns around and gets a reward. Hits the correct light, turns around and gets a reward. Then he does this part of the experiment where he anesthetizes the animal and then wakes him up by stimulating his ventral tegmental area again. The animal's going to wake up. When he wakes up, he's actually going to go back to work. Now, when we do this, though, we don't turn the anesthesia off. So whatever's keeping him awake is actually overpowering the anesthesia, so much so he's actually cognitively intact and is actually performing. Then when Ken turns off the stimulation, in effect, the anesthetics are going to take over again, and the animal's going to go out. So let me just show you this. So here's the animal with no stimulation. He's anesthetized with isoflurane. And Ken's going to turn the stimulation on now. So he turns it on. Gets up, looks for food, but he has to work first. He has to execute the task correctly. So he comes over, hits the light, and gets a reward. So the anesthesia is still working, right? So whatever's working here is actually overpowering the anesthesia and driving his brain, right? Hits the correct light again. He's a little wobbly, but the point is he's cognitively intact. He knows what he's supposed to be doing. He's actually executing the task. Now he's going to turn around again. I think this time he gets this one wrong, gets this one incorrect. So he hits the light tells him it's wrong, so he won't get a reward this time. Now watch what happens here. He turns around one more time. Going to turn around one more time. And Ken turns off stimulation and he's out. So what are we doing here? So here's the ventral tegmental area here. It's sort of the starting point for the mesocortical pathway, meaning midbrain cortical pathway going up to the cortex through the limbic system, amygdala, hippocampus, nucleus accumbens. And this pathway is thought about mostly for reward, but it's also an important cognitive pathway because when you administer Ritalin to treat ADHD, this is the pathway that you're actually affecting primarily, as opposed to, let's say, striatum in the basal ganglia over here. So this is the way Ritalin would work. It would block the reuptake of dopamine, and it has this cognitive enhancing effect when you take dopamine. When you take Ritalin. And so Ken has done the same sort of experiments by administering Ritalin instead of stimulating directly. What I showed you here is he stimulated directly electrically, and he's also done it optogenetically, stimulating the D1 neurons, and you can actually get the same effect. So something in this class of drugs that stimulates or arousal-promoting drugs is going to work, and we want to bring it to market so that it can be brought into practice, because this would change anesthesia practice dramatically, because 25% of kids coming out of anesthesia with the inhaled drugs will have brain dysfunction. And certainly elderly patients, you can almost count on 20 to 40%, depending on what study you want to look at. So we should make this a new part of our practice, which is turning the brain back on after we've kept it anesthetized so profoundly for so many hours. And so this is what I've just shown you. The brain in anesthesia is dynamic. It's not turned off. I've shown you something about the mechanism of action. It's by creating these oscillations, sustaining them, and how they can impair. We got some idea of how, particularly for propofol, how the oscillations correlate quite tightly with the change in spiking activity in the cortex, across the cortex, basically. So you can develop monitoring strategies, because these changes in the oscillations and the actual oscillations themselves are not subtle, they're very obvious, and they change in very systematic ways with the anesthetic class, the dose of the drug, and the patient's age. And so closed loop control I showed you is very feasible. It's not something which is currently done. It's never been done in the United States. It's been done in Europe, where they use much more intravenous anesthetics, but it's certainly not even there. It's not part of standard practice. But if you did closed loop control and you turn the brain back on, this would be a way of speeding recovery, giving patients just the anesthesia they needed, no more, and hopefully preventing this postoperative cognitive dysfunction. One of the things I'm just suggesting here is that, which is sort of what I've learned through my research, is anesthesia sits here, and it's strongly connected to a lot of other problems in clinical neuroscience. And we haven't made these connections as robust as we probably could have, or as we probably should have, because we don't think of anesthesia as part of neuroscience, we think as part of pharmacology. But yet, I mean, think about it. Like ketamine, for example, is a very potent pain reliever. It's also used to treat depression. It's a model for schizophrenia. Epilepsy, you use, as I said, high doses of the anesthetics to actually treat epilepsy. At low doses, alfentanil and also methotrexate can be used to actually induce seizures. If you're in the operating room and the neurosurgeon's having trouble finding seizure focus, you can give some alfentanil and actually locate the seizure focus. There's a strong correlation between the way certain animals hibernate and deep anesthetic states in terms of the actual physiology of the way GABAergic anesthetics work. It's very much the way that certain animals like the painted turtle go into hibernation. And the ideas we've been talking about here for turning the brain back on under anesthesia could also be applied to enhance coma recovery. So by trying to bring anesthesiology research more to the neuroscience fold, we should be able to take better advantage of ideas which are coming out of these areas of clinical neuroscience research and basic neuroscience research, and also contribute in the other direction as well. And ultimately, our patients will benefit from it. So thank you very much again. It's a pleasure. Sorry I couldn't be there in person.